So, get this. What if you could break a fundamental law of physics just with a piece of software? Sounds like science fiction, right? Well, today we're going to look at a totally revolutionary algorithm that's doing just that. It's smashing a barrier in microscopy that's been around for almost 150 years, and it's completely changing how we see the very building blocks of life.
Okay, so for generations, scientists using light microscopes have run smack into a wall. It's this hard physical limit where, past a certain point, everything just turns into a blur. But what if the key to punching through that wall wasn't some crazy expensive new lens, but just a smarter piece of code? Right, so the villain in this story, the big bad, is something called the diffraction limit. Just think of it like this. When light goes through a lens, its waves kind of spread out, like ripples in a pond. Now, when you're looking at something incredibly tiny, that spreading effect creates blur. A physicist named Ernst Abbe figured this out way back in 1873. He calculated that because of this blur, any two things closer than about 200 nanometers would be totally impossible to tell apart. They just melt into one single fuzzy blob.
And for more than 100 years, Abbe's limit was pretty much a hard and fast rule, a wall you just couldn't break. Then, in 2014, a Nobel Prize was actually awarded for these amazing techniques that found clever ways to get around it using some really complex hardware. But then, in 2022, something totally different happened. A new path opened up. The MSSR algorithm was published, and it showed a way to get that same super resolution using nothing but pure math, no expensive machines needed.
So, let's talk about those older solutions. Now, don't get me wrong, those Nobel-winning hardware tricks were absolutely incredible breakthroughs, but, and this is a big but, they came with some serious trade-offs. Getting that extra bit of clarity was not simple, not cheap, and definitely not fast.
And you can see here the tough choices scientists had to make. I mean, some methods would physically mess with the light itself, but that meant you needed these incredibly expensive, super specialized microscopes. Other ways involve painstakingly locating one single molecule at a time. This could take thousands, even tens of thousands of images, way too slow to watch anything alive. So it was always this tough compromise. You could have it cheap, fast, or good. Pick two. Okay, this is where MSSR just completely changes the game. It stands for mean shift super resolution, by the way, and the genius of it is that it doesn't try to change the light or the lens at all. It just takes the blurry image you get from a regular everyday microscope and makes it sharp using pure computation.
And here's the core promise, literally right from the title of the research paper. The absolute game-changing idea is getting that super resolution from just a single picture, a single frame. No more waiting around for thousands of images to process, no more dealing with these massive, clunky datasets.
So, the really crucial point here is that MSSR makes super resolution accessible to pretty much everyone. You don't need a $1 million microscope anymore. Nope. Any lab with a standard fluorescence scope can just download this free, open-source tool and start smashing that old diffraction barrier. Oh, and as a little bonus, it even cleans up the image noise. It's basically the democratization of seeing the impossibly small.
Okay, so how on earth does it actually work? You might be thinking it's some impossibly complex math formula, right? But the magic here isn't that. It's a single, really elegant insight. A clever trick, really, that just completely flips the problem on its head.
So, the old-school way to find the source of a blurry spot of light was to, well, look for the brightest point, right? The very peak of the blur. But the problem is, the top of that peak is kind of wide and fuzzy, which makes it really hard to know exactly where the true center is. MSSR just throws that whole idea out the window. It doesn't look for the peak at all.
Here's the logic behind it. The algorithm scans that blurry spot of light, okay? But instead of trying to find the brightest point, it calculates something called a mean shift value for every single location. You can just think of this as a measure of how fast the brightness is changing from one point to the next. And here's the secret sauce. Right at the true, perfect center of the original light source, that mean shift value drops all the way down. It creates this mathematically perfect valley.
And this, right here, is the aha moment. The whole problem was this wide, fuzzy peak of light. MSSR's calculation basically takes that fuzzy peak and transforms it into a perfectly sharp valley. So by finding the absolute bottom of that mathematical valley, the algorithm can pinpoint the true center of the light source with just incredible precision. It's like turning a fuzzy mountain into a sharp little pinprick. Okay, so that's the theory. Sounds pretty cool, right? But what does it actually look like in practice? Well, this is where a really clever piece of code produces something, and I mean this, truly spectacular.
And here's the proof in the pudding. A standard, pretty high-end microscope can see details down to about 240 nanometers. Anything closer than that just blurs together. Now watch this. You take that exact same blurry image and just run it through the MSSR algorithm. And boom, suddenly you can clearly see details that are only 120 nanometers apart. That is a 2 times improvement in resolution, instantly, with zero new hardware. It's amazing.
But what's even more incredible is that MSSR isn't just for standard microscopes. It can act like a booster shot for images that are already super resolved. So for example, you might have a super powerful STED microscope that gets you down to 90 nanometers, which is awesome. But then you take that image and process it with MSSR, and you can push the resolution down to an absolutely astonishing 20 nanometers. It literally enhances the enhancer.
And it doesn't even stop there. The potential goes even further. While its biggest advantage is working on just one single frame, MSSR can also analyze a whole movie, a series of images over time. And by combining all that information from multiple frames, researchers have been able to push the experimental resolution all the way down to just 40 nanometers. I mean, that is sharp enough to see tiny little structures that were once completely and totally invisible with a light microscope.
So, yeah, the pictures are incredible. But what does this really mean for science? Why does a sharper picture matter so much? Well, as it turns out, the impact is profound.
This right here is why MSSR is such a game changer. Because it makes super resolution so cheap and easy, it just throws the doors wide open for new research in labs all over the world. And because it works on a single frame, scientists can finally study really fast-moving stuff in living cells at a resolution they could only dream of before. We can now actually watch the machinery of viruses, see DNA coiling up, and observe the dance of molecules, all of it, in real time.
And scientists are already putting this thing to work, you bet they are. They're using it to watch viruses actually build themselves, to observe all the dynamic things happening inside plant cells, to track the movement of chromosomes. I mean, the core idea is so powerful that some researchers are even looking at using it for astronomy. Imagine that, sharpening our view of the entire cosmos.
And, that leaves us with a really fascinating thought to end on. What MSSR really shows us is that sometimes the biggest leaps forward don't come from building bigger, more expensive machines. They come from just a new way of thinking about a problem. So if one clever algorithm can tear down a wall in physics that stood for 150 years, it really makes you wonder, doesn't it? What other invisible worlds are out there just waiting to be revealed by the next clever idea?